Jagtap, who is also the co-leader of this shared resource called the Proteogenomics Shared Resource. So I guess we were talking with Aaron Schultz about this. This is sort of the kickoff of this as a full shared resource, because in the prior Cancer Center funding period, we were a developing shared resource, but I'm happy to say we made the cut and we are up to a full-on shared resource. So I guess we should have streamers and things, because it's the kickoff as we enter that next phase. Just quickly, before I even get into any of this, and I know Pratik will have a few of these acknowledgments as well, what I'm going to be presenting here is, as you're going to see, a lot of work developing sort of a bioinformatics platform, so to speak, in this area of multi-omics and proteogenomics. It's been done through a number of years, through numerous collaborations, so it is a lot of people have contributed. I've kind of highlighted here in bold a number of people who I'll at least touch on some of the things that they've done, but both here at the university, people who we've worked with who are actually using these in projects, as well as a whole slew of people from around the world who develop various software platforms that we've integrated into what we do as well as a number of funding streams. One of those that really kind of helped get this off the ground is through the NCI, and that is the Informatics Technology for Cancer Research program that really helped push this forward from funding we had a few years ago. So just a quick touch on some of the people who have actually made this possible. So outline, what are we going to talk about? So our hope here is today is to mostly give you just a view of kind of what we do in this shared resource, and I'm going to kind of say, you know, what is this proteogenomics thing, and try to give you a sense of that. We're going to kind of not go real deeply into any one project, but kind of just give you some snippets of sort of what is it we do, what are some of the applications. And we'll go pretty quickly, so I know probably some of this might go past you pretty quickly, but hopefully if some things catch your eye as a researcher doing various projects and it looks of interest and maybe of value to something you're doing in your work, you can follow up with us and we can have some more discussions about, you know, what could we do to potentially help your work along. And we've kind of got this divided in two segments. My part will be what we would call, I guess, more traditional proteogenomic applications. And then Pratik's going to talk about this thing called metaproteomics, which is sort of a branch of proteogenomics, but more about how do you utilize some of these tools to look at and characterize microbes and microbiomes and their role in cancer and disease and things. So that's how we're gonna divide this up. And as just kind of a first sort of intro here again, what is the shared resource, this proteogenomics shared resource? And I know all along we've been trying to kind of help define what we do. So this is an attempt at kind of telling you what it is we actually do. And really what it is I would say specialized bioinformatics tools that we offer and offer sort of a full range of consultation, development, application, training, all of those things that go with it, but really centered around this integration of biological mass spectrometry data, primarily proteomics, but also we do some metabolomics work, so we have some sort of talents in that area as well. And then integrated with other levels of information such as genomic and transcriptomics, and you'll see that as we go a little bit. One thing is we are not a data generating shared resource. So we take in data that comes from like analytical biochemistry, from mass spectrometry of proteomics, whatever that type of data. I also direct the Center for Metabolomics and Proteomics here, which is another data generating center, as well as things like cancer genomics, people doing the RNA-seq and the DNA sequencing. They're generating the data. We are helping bring that together and hopefully answer important questions in cancer research by analyzing this in sort of customized ways. And as I said, really what we are is people who have the expertise to actually make that happen. And I down here have Pratik and Subina. Reed actually works with us through the Supercomputing Institute, and I should probably give a shout out to the Supercomputing Institute, because they really are the ones that provide us the infrastructure to support the software and the storage of data and really help to keep this maintained. And as it says here, what this really is a very collaborative type of arrangement where people have the right types of data. They're trying to answer questions. We act as consultants as well as the people who say, okay, let's design these experiments in a certain way. Let's take that data. Let's figure out what are the tools that are needed to analyze and integrate that data, help apply it to their data, get answers and to their questions, as well as we do a fair amount of training, trying to say, okay, we've tried to develop these tools in a way that it's not just us that use them, but others. So how do we train people in their laboratories to kind of get up to speed? So now we can sort of make this more efficient. You get your data, you pump them through these tools, you get your answers, generate your next hypotheses, and off you go. So it's a little attempt to kind of explain kind of how we operate and what we are. Okay, so into the sort of a little bit more of the science, and I'm going to, as I said, kind of talk here to start about traditional proteogenomics. This is just my sort of calibration slide because I know not everybody kind of knows the ins and outs of proteomics on a mass spectrometer, and that's what we're going to talk about here is sort of the integration of proteomic and genomic data, proteogenomics. This is kind of that first starting slide. So what we're talking about here is the traditional bottom-up or shotgun proteomics, where we take complex mixtures of proteins, we turn them into peptides via trypsin digestion. We put them through a mass spectrometer where we record the masses of all the peptides as they come off a chromatography column. We try to then isolate each of those peptides that come through into the mass spectrometer. That's what we're showing here. This would be a single peptide. We fragment it into amino acid pieces. That is sort of a fingerprint. We detect the masses of those pieces that go with that peptide sequence, gives us sort of a fingerprint of that sequence. And then we attempt to say, okay, we now have this sort of unique sequence with this fingerprint of masses that go with whatever its amino acid sequence is. Now we try to annotate that and say, okay, what is the sequence that belongs to this particular pattern? This is where we have to take it through some computational steps, some software. This is sequence database searching, where we say, okay, here's all the known proteins that we think is in this organism that we're studying. Take all those proteins, generate their theoretical spectra that would go with all the peptide sequences that are a part of the proteome. And then based on what we measured here, do a match to that and say, okay, which of these predicted sequences in the database match what you actually recorded in your mass spectrometer. And when things all work well, we actually get an amino acid sequence that matches to that fingerprint, that pattern. We give that a name then and say, okay, this is the sequence. And then we can infer back to which protein in our starting database does that sequence belong to. So there's a whole inference back to the proteins, and that sort of builds back up which proteins are there. There's a whole number of methods that can be done for quantitative proteomics, where you can now look at the different levels of the proteins that have come through that you've identified and start to answer interesting questions in biology. So proteogenomics kind of takes a little bit of a twist on this, and this is just a really basic slide that kind of gives a look at that and says, okay, if we think we need to do this matching to a database of known proteins. So yeah, you can take, your reference proteome that you know for humans or mouse or whatever it might be. But we also know that's not going to necessarily capture all of the intricacies of your particular sample, especially if we're working in cancer samples where we have mutations at the DNA or RNA level that are changing, the nature of the transcripts that can then get translated into proteins, which may also give you novel protein sequences that nobody's seen. It's not in a reference database. So how can you make this more customized? One of those ways is to utilize next generation sequencing in your samples, and generally this is done by like RNA-seq. Now you take your sample, you generate this whole profile of transcripts, you take those transcript sequences and you predict out proteins that would be translated from those as possible proteins, and that's now going to include novel events that are happening. And now you use that as your database to match your mass spectrometry data to. And what happens there, without going too deep into this, is that when you now take your sample, that same sample you have, say, done the RNA-seq on, you do proteomics on it, you generate all these fingerprints of peptides. Now you use this more customized database, and you're now starting to match to novel sequences that are in your sample. And then you can map these back to the genome and start to answer questions about, well, what is this novel sequence? What's the nature of it? Is it coming from some region of the genome that maybe wasn't even thought to be transcribed and translated? Is it in a coding region but happens to be, say, a splicing variant that we're picking up at the amino acid level, or a single amino acid change, or a short indel, whatever it is? You can now start to map these back and actually get the power of this, and this has been used by the National Cancer Institute as an approach, is now you have protein level evidence of these changes that are happening that is beyond just predictions from the genome or the transcriptome. So it gives you a more direct analysis of those proteins that might be doing something, might be causing cancer, biomarker, whatever it is. So the challenge here then is that it really kind of adds all these layers bioinformatically of how do you do this and do it in sort of a routine way, because now not only are we having to take genomic data and create these databases, but now we're going to have to pull in the mass spec data and match them to this new database. And we're going to have to take it through a lot of steps of filtering this and making sure, are we being careful? Is this real? If we think we really are seeing novel variants, how do we know these aren't false positives? Whatever it is. And then trying to interpret what that actually means. So that was the challenge that we were faced with a number of years ago as we started getting interested in this type of work. So what we turn to is to, how do you do this, but also how do you do this for, truthfully, a lab that we're more biochemists. I'm not a computer scientist. I wanted tools that answered my questions, but also could be kind of accessible. So we didn't all have to be programming and sitting there and doing our command line stuff and writing our own little software just to do what we wanted to do. But could you make people like me who don't do that some sort of an interface where I can understand it, use it on my data, get results, and say, hey, this is interesting. Now I'm going to follow this up with new experiments. So what we turn to is the Galaxy platform. Many of you probably are familiar with that. It's been used heavily for genomic DNA sequencing, RNA sequencing software, a platform that brings together all this software to help you analyze complex biological data. But it's open to basically any types of software. So it's really a sort of workbench where you can integrate various pieces of software and make them work together to answer questions when you have complex, and in this case, complex multi-omic data. So we started in on a project that we called Galaxy for Proteomics, or Galaxy P, because the Galaxy sort of platform at that point had all kinds of great software, but it was generally all about genomics and things. So we started to pull in proteomic software and start to talk about how do we integrate these together, doing this in ways that we also didn't want to have to reinvent the wheel and say, well, we got to from scratch build all of our own new software, but bring in all kinds of different software packages that are really high value, but integrate them together. So that's where we have kind of been working, is in this space of trying to develop these tools in a platform that we can then also push out to others. A big part of Galaxy is they have, I think, probably the most powerful training resources for training people on how to use these. They have a whole training network, which we use quite a bit. So it had all kinds of nice features that we kind of led us in that direction. This is just another view of Galaxy in terms of this resource, this platform that can run on HPC, so high performance computing with lots of memory, lots of storage, so you're not having to do this all on your laptop as well, but a platform that can pull in your data, pull in all the various tools that you need to analyze, interpret, visualize, put this onto a server that you can now remotely access, so you don't have to worry about, oh, my storage is done, I don't have enough memory on my little laptop here, and do this all in a way that biologists really that are at the bench can learn this, use it, has an interface that's usable on the web that you can start to learn and is pretty intuitive, and pull this all together to make it work. So that has really been kind of our approach to things, is how to develop Galaxy to use this for our own needs as well as try to push this out to others. So without getting into every piece here, I'll just kind of give a few snapshots of kind of how these workflows kind of go in the proteogenomics realm. So this challenge in this case of integrating RNA-seq and proteomics, this just kind of outlines sort of our current workflow, which we developed a number of years ago, but is still very useful of, in yellow, bringing in these different pieces of raw data or databases that are like reference genomes and things where we take the RNA-seq through a number of tools, and those who work in this area will recognize these are a lot of the standard tools everybody uses to assemble RNA-seq, pull out variants and things along those lines. And then also pull in the mass spectrometry data from the same sample that we're trying to analyze potentially, and now go through all of these steps to ultimately make these novel protein sequence databases, pull in the mass spectrometry data, push that through and start to identify potentially novel amino acid sequences that belong to express proteins in your samples. So this takes a lot of care. And this is a figure from a paper. We've had some collaborations with Natalia Trechakova, and this is Andrew Roszewski and some of his graduate work. But we've taken a lot of care in also saying, okay, when we do find, say, novel amino acid sequences, how do we make sure these things are not just some false call to something and we're chasing ghosts here and this isn't really real? And just I'll highlight here is that we've put in a number of tools on the back end. So if you get matches to peptides that are potentially novel sequences, we take them through a couple more layers of analysis where we do a BLAST-P step to say, okay, is this really truly not a reference known sequence that we've identified? And also other tools, there's a tool called PepQuery, which also does a further sort of evaluation of, you really think this spectrum matches this particular variant sequence? We're going to do a rigorous reevaluation and ask, is this there's some other answer here, a PTM to that sequence that's actually a reference sequence that actually matches it better? Is this really true is what we're always asking. And if they pass all of those steps, then we can sort of get to a point where we say, okay, we think this is really a variant sequence that should be considered. We've also developed a number of tools then on the back end of, how do you sort of interpret and visualize this? And without going into much detail, this is a tool, this multi-omics visualization platform, which gives you the ability to say, hey, we identified in this case a single amino acid change in a protein. Let's map that back to the genome, and it's coupled with the IGV viewer where now you can sort of bring this back to the genome and look at, at the genome and transcriptome level exactly which gene was this and where did this change in a nucleotide happen that ultimately introduced in a single amino acid change. So trying to get some tools that connect all of this together to visualize things, as well as some ways to like interpret what you're seeing. What are the sort of meaning of this, which is the bigger question, right? So great, I found some switched amino acid sequence and some protein that I'm interested in. Does that mean anything, right? So this is at least a tool. This was a collaboration, this Cravat tool, which was focused on genomic sequences where you have your RNA-seq and you put it through this and it does this sort of impact analysis of, where in your protein is the change happening? What might that do structurally? Where are PTMs in that sequence that could be changed because of this, whatever it is, as well as things like network analysis. And we modified that so now you can take protein level information on it's an unique amino acid sequence, relate that back to the genomic coordinates, and then do this sort of impact analysis as well. So trying to give sort of a complete toolbox to people to try to interpret this. But ultimately, what we want to do is then give them good leads that they can go back into their experimental system and try out this mutated protein potentially. Does it have a functional role? What does it do? Those types of questions. So we only can go so far sort of bioinformatically, but we hope that kind of pushes the research along. Another thing that is really common that people want to do is you've got quantitative proteomics data, you've quantitative, say, RNA-seq data, looking at levels of transcripts that are under different conditions. Let's compare, how do these things respond at the proteome level versus the RNA level? So you start to look for mechanisms and potentially gene products that are regulated post-transcriptionally if these things don't match up well with each other. So this has been done for quite a while. This is another figure from Andrew, but that says, let's compare the RNA expression levels and how they change their dynamics versus how do the proteins, the corresponding proteins that are encoded by those transcripts, how do they change? Do they look similar? Do they look dissimilar? And what does that mean? So we generated and came up with a tool that works within Galaxy to do this in a relatively straightforward way, where it takes in the RNA-seq data, takes the quantitative proteomics data, pushes them through, and now gives you kind of a slew of different outputs that help you visualize these differences, as well as sort of characterize, well, which genes, which RNA molecules and proteins that are corresponding are the most dissimilar that I might focus in on, and things of that nature. So this is a software called QuantiP, this sort of interactive visualization tool. And I put this in here just as another, there's actually, this isn't work of ours, but there's a group that's proteore.org, it's called. This is a Galaxy-based platform, but has pulled in a lot of functional analysis tools for visualizing enriched pathways and functions, like through Reactome, but a really simple to use interface in Galaxy. Gives you a really nice ability to say, and I know this is small, but if you have some data set of proteins, and what I'm actually showing here is metabolomics data, which you can map onto these, some sort of output that you can map onto these functional pathways, gives you a really nice visualization of what's being enriched in your samples. This is from an example from, we're working with Bruce Blazer on some work, and this is actually metabolomics data. So it can cross into that omics realm as well. But there are definitely tools for doing sort of pathway enrichment and functional analysis of whatever the outcomes are from these workflows. The last few slides that I'm going to show here are just a little bit of some ongoing work that is kind of I guess a twist on proteogenomics, but a really important one, and that is in this area of neoantigen identification using, I think this is the best term for it, truthfully, immunopeptidogenomics. So we're not looking at now proteins, but we're using these tools to ask, okay, if there's novel transcripts in your sample that are turning into novel protein sequences, potentially those are recognized as foreign sequences, presented on the MHC complex, the immune system reacts to them, et cetera. So this ties into all of the very exciting and expanding work in immuno-oncology. Can you identify these antigens that are novel for tumors and things, and potentially use these as targets and other things in cancer research? So there's this whole workflow that has been developed, not by us, this has been around for quite a while, but where you can now take your samples and you can enrich those MHC, whether it's 1 or 2, you can enrich those complexes and the peptides bound to them, put them through a mass spectrometer and say, okay, which sequences are being presented on the MHC complex that we may be most interested in. And so you get this output of it now identifying these sequences. The trick here, though, is if you really want to look for novel things, again, you got to integrate in, say, RNA-seq data that says, well, what are the possible novel sequences that we're trying to identify, right? So it's kind of fits into this proteogenomics flow. Really complicated slide. The piece here that I think is key, though, is that if you really want to do this comprehensively, right, you have RNA-seq and you can now start to ask, what am I interested in? Am I interested in splice neojunctions? Is that in my cancer the main kind of player that I'm going to look for in these neoantigens? Am I looking for short indels and other things? Am I looking for structural variants, things that are changing the genome and then turning into these structural variants, transposable elements? So it's challenging because there's a lot of avenues to get these neoantigens, and so you need the tools to try to do that from the starting RNA-seq data. And so Galaxy can give you those options, which is a really nice feature, and also do things like predict which of these novel sequences might actually bind the MHC. So there's a whole computational workflow there. But in the end, what you then generate are prioritized potential neoantigen sequences that you can now couple together with the workflow, where if we generate that peptidomics data in the mass spec, we can then match those back to this database, use some of our tools to really be careful about, well, which of these are real, and ultimately then output potential verified neoantigens that we have enriched out of our sample that are actually being presented on the MHC complex, and you might want to follow up on in whatever the study is. We have an ongoing study. A lot of this has been pushed forward with Dave Largaspada on a grant that we're a part of, looking at these malignant peripheral nerve sheath tumors. And this is just a snapshot. These are these peptides that we've identified from some of the work that are these tumor-associated antigens. So these are actually reference sequences, but are known to get presented in tumors and in cancer that also can be identified using this approach. So it has a lot of its own features here. As you notice, these are almost always like 8 to 10 amino acids, so they're short peptides. They aren't tryptic peptides because now we don't digest these. We just take straight what comes off of the MHC complex. So without going into detail, that offers actually a lot of computational challenges because we were not used to looking for those types of peptides. So, but it is definitely something we're continuing to work on and has a lot of value and power. Okay, so that's my half here. I'm going to hand this over to Pratik, and he's going to take you home with sort of this idea of metaproteomics and what is that and how can it be useful for cancer research. So it's all yours.
Thank you, Tim, and thank you everybody for coming in on a cold morning. I'm going to switch gears a little bit now. I'm going to talk about proteins that are expressed by microbiomes that are present in clinical samples. So, Tim kind of gave a presentation about proteogenomics, which detects novel peptides or proteins that are associated with variants that are present in samples. But I'll be kind of talking a little bit more about how does microbiome kind of affect, or how can we detect microbiome changes in a clinical sample? So, I just kind of wanted to introduce myself. My name is Pratik Jagtap. I work with Tim. I'm part of the Galaxy P team and have been the co-lead on the Galaxy P team for last 11 years now. Just wanted to give a brief overview of microbiome research, and I'm sure most of you might be aware about this, especially in the area of gut microbiome research. There has been a lot of interest in understanding how the gut microbiome affects the health of an individual, both health as well as disease state of an individual. And then there has been also a lot of work that has gone on to show that that the microbiome could also be involved in cancer progression. So some microorganisms have been implicated to have a role in cancer progression. There have been also a lot of challenges that exist in not only trying to correlate the presence of a particular organism to cancer, but also trying to find out what does this organism actually do in cancer, right? And that's where we think we can bring in some tools, some methods, such as metaproteomics, which I'd be introducing to you, which not only looks at what are the organisms that are present in the sample, but also predicts or kind of even finds out what are the functions or what are the proteins that are expressed by these microorganisms. And hopefully that helps in finding out a little bit more about the mechanism in which these microorganisms interact with the host. So most of the microbiome research uses metagenomics, which is study of the DNA that's present in samples, and that definitely gives you a good idea about what's the taxonomy that taxonomical composition of these microbiome. There have been some researchers who have started working on metatranscriptomics as well. What we have been working on is an area called metaproteomics, wherein we look at proteins that are expressed by these microorganisms. And one of the advantages with this is, as I said earlier, it tells you about what are the functions that are expressed, and hence gives you some idea about some mechanistic details on how these microorganisms might be affecting the host and its environment. So to give a very brief overview about, and again, this slide is about gut microbiome, but this can also be used for other clinical samples. So for example, you start with the sample and then you perform protein extraction. So you extract proteins from these samples. And once these proteins are extracted, just like Tim mentioned in his proteomics mass spectrometry workflow slide, You digest these proteins, subject them to separation, identification, and quantification using mass spectrometers. And then once you have the mass spectra that are generated, the fingerprints that Tim mentioned, you can perform a database search against not only a database with your host proteins or host proteome, but also microorganism proteins that are present in this database. What this basically leads to is you end up identifying microbial peptides, which you can use for taxonomic composition, or deciphering the taxonomic composition, and microbial proteins, which kind of help you to understand the functions that are expressed by these organisms. So the term metaproteomics actually was coined almost 20 years ago now. Just to give a very brief overview, in most of your metagenomic studies, you might actually end up detecting a similar taxonomy, but if you subject it to different conditions, so let's say you offer the same set of organisms, different nutrients, you end up getting different functions, but those are not something that are captured by metagenomics. But we think that through metaproteomics, we can capture that. You could also have another scenario wherein you have totally dissimilar taxonomy, but you kind of you treat them with the same kind of nutrients. You should see similar proteins or functions that are expressed because similar enzymes that are expressed, and that should be captured by metaproteomics. So we think that the power of metaproteomics lies in not only finding out what's the taxonomy, but also how these organisms are reacting to the immediate environment. So one of the challenges with metaproteomics, especially in the clinical field, has been that in most of the studies, and these are 3 of the studies that we have conducted, one of them, 2 of them which have been published, and one of them is getting reviewed. We find that most of the peptides shown in this orange slices, microbial peptides that are detected, are a minority. It's a really small slice in the number of proteins that are detected, and most of the proteins that you end up detecting are the host proteins, right? So yeah, actually getting a lot less information that you have to make some information to make the most out of in your analysis. So with this in mind, we have developed a workflow, not necessarily to enrich the microbial content, but to try to capture as much microbial information that is possible. And I'll be going through this workflow a little later. I just wanted to show you that this particular workflow helps us, which has got multiple tools, helps us to not only detect multiple microbial peptides, but also verify these microbial peptides and eventually quantitate them and use them for microbiome studies. So Tim has kind of gone through this earlier. So we have used Galaxy interface for having all of these tools within these workflows. And again, this Galaxy interface for the researchers of University of Minnesota can be accessed through MSI. So there's galaxy.msi.umn.edu, which is a website that you can use. And if you have a MSI login, you should be able to access these tools, not only the genomics tools, but also the proteomics tools that we have developed. One of the advantages of Galaxy is that not only access to tools, but you can also view in tools or kind of have tools in a particular succession so that you can go from point A to point B, as has been shown here. You can kind of integrate these tools into a workflow and hence get your outputs. Using these workflows, we have published quite a few manuscripts, including some of the research in human salivary supernatant with biofilm models of dental caries, with cervical vaginal microbiome, along with Amy Skubitz, some of the work with Manish Bhargava in the bowel fluid metaproteome, and also while the pandemic was ongoing, we also kind of published a workflow to detect co-infecting organisms that are present in COVID patients. So just to kind of give you an example of one of the work that we have been doing, one of the projects that we have been working on, and this is with Amy Skubitz's lab on detection of ovarian cancer. And this study has been led by Subina Mehta. So ovarian cancer, basically, there are no reliable early detection methods. And so with that in mind, and also the fact that the dysbiosis during oral cancer, during ovarian cancer, leads to a change in the homeostasis between the bacterial communities and the host cell, which could be linked to ovarian cancer. We thought it would be important to detect these host proteins as well as the expression of these host proteins as well as microbial proteins, and try to see if we can look at this expression and integrate this, whether this could actually help in finding out whether this can help in early detection of ovarian cancer. So for this, Amy Skubit's lab had collected a lot of liquid pap tests, and we had many samples of these, and we also had mass spectrometry data for this. So what we basically had was starting with this pap test fluid for at least 20 non-cancer controls and also 20 high-grade serum ovarian cancer patients. So and again, amongst these ovarian cancer patients, we had benign as well as genuine ovarian cancer patient samples. And then this was subjected to a TMT 11-plex multiplex analysis, which actually helps you to use many of these samples together and analyze them together. And then quantitative metaproteomics data was available for this. So the data analysis pipeline that we used here was, we started with these 4 data sets. And again, these are 4 multiplex data sets, so really large data sets. And then we searched this against a large database, a comprehensive protein database, wherein a literature survey was done to find out what are the organisms that are present in, what are the microorganisms that were detected in ovarian cancer. And then we use a tool called MetaNovo to generate a reduced database. From this, we use a discovery workflow, and this is kind of the the heart of the workflow here, in terms of the fact that, since I mentioned before, there are very less number of microbial peptides or proteins that are detected in samples. So we use 3 different search algorithms to detect as many peptides as possible. And then once this was done, we subjected to this tool called PepQuery, which Tim mentioned earlier in the proteogenomics workflow, which looks at, which kind of verifies whether these particular microbial peptides are indeed present in these samples. And this does it in a peptide-centric manner. Once we had that, we selected these microbial proteins that were detected and verified and subjected to quantitative analysis and performed statistical analysis. So this is kind of one of the clinical workflows that we have been using for multiple research projects, which involve clinical samples, but we have also been using it for cancer research as well. So from this study, from the taxonomic analysis, we found that lactobacillus seems to be the most dominant organism, or most dominant microorganisms that are present, or genera that's been present in these samples, followed by bifidobacterium and other organisms. For the quantitation analysis, what we did was, once we had the quantification done on verified proteins, we subjected this to statistical analysis, and this was basically done by biostatistician Ashley Peterson, who's a biostatistician at the Masonic Cancer Center. And what we found was, we could find differentially expressed microbial proteins as well as differentially abundant human proteins, and some of them have been shown here. One of the interesting findings from this study was, and this is again a preliminary finding based on these 4 data sets that we have analyzed, is that when you use performance metrics for finding out whether you can use the data to predict whether you will have ovarian cancer or not, and when you compare the ovarian cancer versus benign patients. So if you use the human proteins data, which has been shown in the blue line here, you do see this performance metric, but it kind of improves if you use microbial proteins as as the quantitative data that is used as an input. But if you use both of them together, it seems to improve the predictive capability of your analysis. And again, we are this is very preliminary finding. We want to ensure that this observation that we have can kind of stands when we do this on a slightly more larger scale. So what we are doing right now is, so after this literature survey analysis, which kind of detected all these proteins, We also subjected these samples to 6-ness rRNA sequencing, which gave us a slightly more expanded or more number of microorganisms, or I would say more customized data, a customized database for metaproteomic analysis. And based on this and the combined analysis of both the database generated from the literature survey and the 6-ness rRNA analysis, we're seeing a lot more proteins as well as microbial proteins that are detected. And we hope that this will actually give us a much better understanding of the data. There are also additional data sets apart from the 4 that we have analyzed, that we are planning to analyze. So we are hoping that we'll get a lot more data and statistical analysis that will help us to perform better predictive analysis using these data sets. The other data set that we have been working on is with oral cancer samples from Frank Andre's lab. And these are basically oral rinse samples that have been taken from patients which have been diagnosed with oral cancer. And some of them have been, collected when before the treatment, and then there was a treatment using a diabetic drug that was used to find out whether it kind of helps in addressing the lesions that are developed because of these cancer samples. And so we have replicates from these data sets, and these data sets were then subjected to a method called as DI passive method, which we kind of sent these samples to Bruker, and they ran this on their timsTOF instrument, and they performed mass spectrometry. And we're kind of very happy to see some of the results that we got through this. So we got a really good depth of analysis. So remember, we talked about the small slice of microbial data that we get through the earlier data set analysis. Now, with this new method, or the method that has been available now, we can kind of go deeper into the microbiome and not only identify more taxonomic species, but we hope that since we are detecting more proteins per organism, we might also be able to find out more functions of these organisms, how they affect the microbiome. So again, very preliminary results. We also hope that we will get some good quantitation. And again, this is from a totally different study, but we had a study on infection of mouse samples, and we collected feces from this. These were infected with E. coli, and the idea was to find out what are the proteins that are differentially abundant. And as you can see, we could find quite a few proteins that interact with which are involved in infection from the host side, and or immune proteins as such. And you can see that they are differentially expressed here. And we also found quite a few differentially abundant microbial proteins. So we are hoping that in our oral rinse or oral cancer data set, as well as other data sets, we should be able to find much deeper insight into microbiome analysis using this method. So to conclude, we've developed a bioinformatics workflow, which kind of takes in clinical samples, and you can start either with a literature survey of your data set, or you could have a 6-ness rRNA data set and kind of generate a much smaller database from which you can detect microbial proteins and then verify them, quantify them, perform statistical analysis. So that's that workflow can also be used for cancer studies, because you can take any clinical samples and use that for and try to find out the correlation of these functions as well as taxonomy with that particular disease that you're studying. For the ovarian cancer study, we have detected quite a few differentially abundant host and microbial proteins, and we are actually performing much deeper analysis on other data sets or larger data set to find out if we can use the predictive value of both microbial content as well as host content to predict cancer or detect some features that we could use for targeted analysis. We think that the DI passive method is really helping us to get deeper into the microbiome as well as the host proteome, and hence will help us to get a better understanding of the host microbiome interaction. And it also gives you deeper insights into the taxonomic and functional composition of clinical microbiome. Just as a kind of few concluding slides. So if you wanted to know more information about some of the Galaxy P work that we've been doing, the published manuscripts can be accessed through that link there. And we have quite a few presentations that we have given in last 10, 11 years now. And then you can also contact us in case you have any questions with the contact information here, including some of the, if you have any project in mind that you would like to collaborate, or if you have any questions of any of the workflows or tools that we have developed, you can also follow us on Twitter or X, as it's called now. Lastly, I'd like to acknowledge quite a few researchers, Amy Skobitz, as well as Frank Andre's lab. We have been working with their lab, and also some of the Galaxy P researchers or colleagues that have been mentioned here. I'd also like to acknowledge some of the work that we've been doing with Bruker. They actually helped us to get some of the data sets so that we can evaluate the data set and understand how deep we can go into a microbiome. So we think that that's something that we would like to see if we can use it in the future. Yeah, there's just a slide to show that we are available for any of the newer studies that you'd like to take. As Tim mentioned earlier, we do not generate data, but we help you to make sense of the data that has been generated and hopefully can give you some biological insights based on either your proteogenomic studies or metaproteomic studies. Yeah, so if there are any questions, both Tim and I will be very happy to answer them. Thank you.
I have microphones. Thanks, you guys. I was thinking, since you have a little bit of time, can you briefly talk about your sort of fee structure? Do you put effort on grants? What's kind of the range? And can you do before or after a grant, or how does it work?
Yeah, so the question was sort of fee structure and how do we keep paying the bills? It's a great question. So it really is a collaborative shared resource in the sense that it really is about working together, personnel working with labs. So we don't have sort of a hourly fee structure because it's just, in the end, it's a lot of back and forth, and it doesn't operate very well as well, here's my data, can you just crank it through your tools and give me some results? I wish it was that simple, but it turns into much more of a sort of back and forth and interactive relationship. So our goal is to support as much as we can through grants and through collaborative grants, which we have several of them that are ongoing, as well as get some centralized support through the Cancer Center. So that's kind of how it works. But the key to that, the help of the Cancer Center supporting us, means we don't have to be like a law firm that, okay, you walk in the door and we turn on all right, we're starting a billing, So we can sort of work with people who come and say, hey, I've got this data, I got an idea. Can we try to generate some preliminary results to put in a grant? So that gives us flexibility to do that so that it's not just sort of like, okay, if you want to work with us, here's our hourly fee, and we'll start the clock ticking when we start working with you. So it's kind of a case-by-case basis, but always the goal is to, okay, let's help generate some preliminary data and let's get a grant funded, and then we can do this in a larger way. And to try to help with our bandwidth and capacity, like I said, the other kind of key philosophy is we do a lot of training, so we sit down and say, hey, we've got these tools already. Let's see if a grad student or somebody in your lab can help learn it. So we spend the time, and that again is sort of supported through some of the central funding, and get people up to speed so we can just say, great, we'll just be here if you have questions, and you start running your data and away you go. So that's kind of how we're trying to make this go.
And can you give us a feel for like how busy you are right now? Are you like you have several groups you're working with pretty actively, and
Yeah.
how much capacity could you take on?
I'd say we have capacity for more always. And that would also, what would help is if we can get grants funded, then we can focus on those projects too. Whereas if we're continually trying to get more funding, we don't have as much capacity to help people. So it's a balancing act. But we certainly have the ability to take on more projects. And like I said, we've tried to develop this so it isn't all on us to be the ones having to analyze everybody's data. It's more let's sit down, train you on how to do it. We'll be your consultants and we'll work with you, and maybe have some effort that's paid through whatever funding stream that's supporting a project, but kind of to the best we can put it on to the other people in the group, and then they can do a lot of this too. So we're open for business, I guess, is what I'm saying.
Great, thank you.
Are there any other questions? Let me just check my virtual audience. So this is a question from David Largaspada. Have you found any useful information in post-translational modifications of microproteome data? And what orthogonal methods are being used for quantification to check the proteomics?
Can I answer that PTM question?
Do you want to? Okay. Yeah, so the question was, in the microbiome data, can PTMs be analyzed, and has that been analyzed? And the other question was, what are the quantitative methods used in proteome or proteome analysis? So to answer the first question, so we have not yet looked at protein translation modifications in microorganisms, because as I said earlier, we are looking at a really thin slice of the proteome, which is a micro, especially in clinical metaproteomics. But we think that as we start using more sensitive methods, or if we start enriching some of these microbial proteins, we should be able to get a lot more representation of these microbial proteins, and hence should be able to see post-translational modifications. But so yes, there is an interest, but we haven't really looked at it right now. Do you want to answer the quantitation question?
Yeah, so I think the question was like orthogonal to, like, you take quantitative proteomics data and you quantify a bunch of proteins, and it says, hey, this protein is up and this down. How do you sort of, in complementary or orthogonal ways, how do you confirm that? So a lot of it's done, standard, more classical approaches where I've found these proteins, I think they've changed. Do I have an antibody? Can I do a Western blot and confirm it that way? Personally, my experience with Western blots is I believe the mass spec data more sometimes, because they're still the gold standard to some degree. Another really effective way to quantify is this idea of you can do two phases of mass spectrometry. You can do this more open-ended discovery phase where you identify things and you quantify them and you say, hey, these are the proteins we think are changing, best we can do it confidently. And you can then develop and follow up using targeted mass spectrometry. So now you've defined the proteins, you define peptides that match to those that are potentially changing. And you can actually use a type of mass spectrometer that you can program to just look for certain peptides and say, okay, let's ignore everything else. Let's rerun maybe an additional cohort of samples or biological replicates, whatever, and look just at the proteins you're interested in. And those are actually quite accurate and quite confident. So it can really give you a verification of your initial results. So it's kind of like a Western blot done in the mass spectrometer where you're targeting certain proteins. So that's one way that can be very useful.
Oh, one more.
Hi, Tim. I have a practical question for you. So is the sampling method the same for metaproteomics versus metagenomics and the storage?
Wow, that's a good question. You want to?
It could be slightly different because, obviously you want to focus on proteins when we want to detect the metaproteome. There have been some labs which have been trying to come out with a kind of a universal protocol where you can use, the same sample preparation method and use it for metagenomics, metaproteomics, and even metabolomics. But I don't know whether there is an accepted protocol that's available that one can use. So yes, if there's a project, we'll be very happy to try that and use it. But right now we have been kind of using these separately, in the sense, even if we take the same sample and subject it to, metagenomics, there's a separate protocol for metaproteomics. So kind of assuming that it's consistent, but yes, there are going to be changes because of the.
And is there a minimum biomass that you need?
So for metaproteomics, you would definitely need more proteins, more sample, because, you, again, for two reasons. One, you lose a lot of sample when you're during the processing, you lose a lot of biomass during processing, but secondly, also the mass spectrometers are not as sensitive. But again, as I said, with these new methods, hopefully we should be able to address that. So yes, I would say slightly more, definitely more than what you would need for metagenomics, but with new mass spectrometers, definitely doable now.
Great, thank you.
I might just add, it's a great question because it is sort of the challenge of not being able to amplify things like metagenomics or whatever, right? So it's always the proteomics issue is we don't have PCR for proteins. So, but we are, one thing we're interested in, and we're trying to get the NIH to fund us to work on this, is how, the magic prep that you'd want to do in these is some treatment that isolates the bacterial and enriches for like the bacterial component and sort of leaves behind the human host component. And then you could really pull those out and get a much more, so there are some ideas about how to do that, but that's where there's a bit of innovation maybe at some point that will come along that will help us prep these types of samples in a better way if you really are doing metaproteomics. But for now, it's sort of case by case, and you do your best to have as much sample as you can, and away you go.
Going back to what Carol asked.
I mean,
If you're running the proteomics score, I have a very good understanding of what the cost is to run a sample or 1,000 through UMGC pipelines.
Yeah.
What's sort of a ballpark cost to run samples in the mass spec for proteomics to generate the data?
Yeah, so if you're going through a core facility, it's going to kind of depend. There's different models in like the cancer center core facility. The core facility that I'm a part of is through biochemistry that many cancer center people come through, is a little different structure because we offer a little bit more end-to-end service of helping with sample prep potentially all the way through generating data and helping like analyze data, et cetera. So, but ballpark, let me think, it depends on what you're trying to do, but a reasonable sized study with a number of samples that you're trying to compare across and get relatively deep, significant mass spectrometry like proteomics data, $5,000 to $10,000 maybe, if it's, and that'll give you a pretty good amount of data to at least find some things that are probably real, and away you go. So, but there's a lot of variables there of what are you trying to do and how many samples. And, yeah.
I wasn't sure if it was going to be 100,000.
Yeah, it scales with, if you're trying to do a study on 200 samples suddenly and it really kind of expands, then things start to expand. But if it's, 20 samples or something, 40, even 50, then, it's in that sort of manageable range. So usually that's workable within a grant application, that you're not breaking the bank on this, so.
all right, let's say thank you for a great talk today.
Thank you.